So hi everyone. Today we are going to go through the secretory pathway of proteins. Okay, it's often asked in a lot of exam questions because usually about 5 to 6 marks because this really tests your understanding in one simple question on the whole process of how proteins are made or synthesized until how they leave the cell. So let's start by where proteins are actually being synthesized, right? So I've already told you before that the ribosomes that are attached to the RER, so keywords here, ribosomes that are attached to the RER, they make proteins. So what happens to these proteins? So these proteins are actually still within the rough endoplasmic reticulum, still within the RER. How do they actually get out of the RER though? So they actually, what happens is actually forms a little vesicle, right? So the proteins are enclosed in the RER membrane. So this is the RER membrane still. The proteins are then enclosed in the membrane, and this, when it pinches off, it forms a vesicle. So why bother forming a vesicle, right? So if you think about it, now we have the proteins that are being made. They are in the RER, they have been moving around, they're made already, then they have to come out, right? They leave the RER. So they leave the RER in a little vesicle. But they're still not complete yet, right? As what we learned earlier, the RER doesn't complete the proteins. It actually needs to go to the Golgi apparatus, right? The Golgi apparatus is this yellow guy over here, right? So the protein in the vesicle now, it comes along and the vesicle fuses with the membrane of the Golgi apparatus. When it does so, the proteins that are found within the vesicle are then released into the Golgi apparatus. So the internal parts of the Golgi apparatus. And this is where, in the Golgi apparatus, this is where the proteins then get modified and packaged. All the keywords are appearing now, right? Modified and packaged. So you add different groups, it'll be lipid groups, carbohydrates, all the different things. And this is where the protein is finally reaches its final form, right? Its full form. And where it is packaged and modified. And then what happens next? We talked about this from the RER. It went from in the vesicle to the Golgi. It goes through the whole process. And now we are at the end here, right? So what happens now? Because all the structures are not connected to each other, and how do they actually move between each other? Can you take a guess what happens next? Of course, it's another vesicle, but this time it is a secretory vesicle. So the secretory vesicle pinches off from the Golgi apparatus, and this secretory vesicle, which obviously also is from the membrane of the Golgi apparatus, this vesicle fuses with the cell membrane or the cell surface membrane. So it fuses with the cell membrane, and in doing so, is able to release these proteins out of the cell. And this is where you get the whole process from the proteins being formed over here until the proteins being able to leave the cell over there.